All right, our new unit is meiosis, okay? It is a form of cell division that we have not really talked about yet. We talked about mitosis, right, where you get two identical daughter cells. Now we're going to talk about meiosis, because this is really what leads us into further deeper into our genetics discussion that we started in the DNA unit. So your objective is to be able to summarize the events of meiosis and explain the reason why haploid cells are necessary for sexual reproduction by completing the formative assessment with the 70%, which shows that you can soar by by integrating information from multiple sources. Okay, so there are three types of cell division, depending on the type of cell, and they are binary fission, which is what like prokaryotic cells do, mitosis, which is eukaryotic cells, and meiosis, which is like sexual cell reproduction in eukaryotes, okay? Recall that prokaryotes, like bacteria, do not have a nucleus, so they simply just kind of replicate their DNA and pinch themselves in half. Eukaryotic cells do have a nucleus. So asexual reproduction versus sexual reproduction. Asexual reproduction is the reproduction of an organism in which one original organism makes an identical copy of itself. Okay? It literally just like splits in half. Sexual reproduction, okay, is reproduction of an organism by combining genetic material from two different individuals of different sexes, right? That's how we all got here, okay? You have to get half of your DNA from your mom and half of your DNA from your dad. That's the way it works, okay? Your mom can't just cut her arm off and be like, grow into a child, arm. It's not going to happen, right? That'd be weird. So why do cells divide? The purpose of cell division in eukaryotes is repair, replace, reproduce, and growth. So cells are tiny, okay? We're made up of trillions of cells. Lots and lots and lots of them. So why not millions or thousands? Why must cells be so incredibly tiny, okay? If cells continue to grow larger, the demands of the cell's DNA increases, and the cell is less efficient at moving waste out and nutrients in. Cell division. So cell division is the process in which cellular and genetic material from one parent cell is divided between two new daughter cells. All right, so here's the parent cell. There are the daughter cells. So each daughter cell has its own nucleus, and the daughter cells immediately enter interphase and begin their own cycles. Pre-cell division. So before the cell divides, the following must occur, right? We already talked about during the synthesis phase that DNA is replicated or copied so that each cell gets a copy of the DNA. And new organelles and proteins must be created so that each cell is equipped for survival, right? Each cell's going to need ribosomes. They're both going to need a nucleus. They're both going to need a Golgi apparatus, you name it. Chromosomes. So chromosomes are located in the nucleus of eukaryotic cells. Okay, and the chromosomes are made up of DNA, which was our whole last unit, right? We talked about DNA and everything about it, and it was so awesome. So the chromosomes are actually DNA and some proteins that it wraps the DNA around to really condense it, okay? And like we learned in the last unit, a gene is a segment of DNA that codes for a particular protein, and it's located on the chromosome, right? When we say codes for a protein, we know that means when the DNA is read and makes an mRNA, it goes out to the ribosome, it matches up with the tRNA, brings in the amino acids, that whole step of protein synthesis, right? That's what we're talking about. So chromosomes are made up of DNA. Histones are proteins that keep the DNA tightly coiled. So what is a chromosome? Typically, when you hear the word chromosome, you automatically think of something like this, right? That's probably the pictures you've seen in the textbooks for years on end, okay? What you are looking at is more specifically referred to as sister chromatids, okay? These are actually two copies of the same chromosome that are attached in the center, okay? Chromosomes can vary in form. They can look like a chromatid, which is just one half, the sister chromatids, which are like hugging each other in the middle, or if it's chromatin and it's getting ready to be copied or read or something like that, then it'll be all uncoiled in like a hot mess, like ball of yarn style, okay? So the centromere is a center of the chromosome. It also serves as the point where the sister chromatids attach to each other. Chromatin is loose, dispersed, immature chromosomes. The cell's DNA is in chromatin form for most of its life. Okay? We have already learned this cell cycle. Yes? Nod your head if you remember this, kind of, sort of. Okay? Cells have very specific time steps to the cell. So you go interphase, and then mitosis, which is our PMAT, and then cytokinesis. Okay? So everything we've been learning about up until now has been about somatic cells, okay? That's what we've been studying. Somatic cells are body cells, or every cell in your body except eggs and sperm, okay? So heart cells, brain cells, liver cells, bone cells, skin cells, those all undergo mitosis, all right? That's really important to realize. Mitosis is the process we have been talking about. That applies to all of your regular body cells or somatic cells. Gametes are what we call sex cells, sperm or eggs. Okay? And gametes undergo meiosis. So diploid cells. Normally when we're talking about our somatic cells, we're talking about diploid cells, okay? This is a cell or an organism consisting of two sets of chromosomes, one from your mother and one from your father. So all of your somatic cells, like your skin and your heart cells and everything, are diploid, okay? Diploid means that it has 46 chromosomes total. 23 from your mom and 23 from your, exactly, dad. Okay? And we symbolize this as 2N, meaning there's two copies of all the number of chromosomes that there are. The prefix di means what? How many? 2, okay? That's why it's diploid, okay? Haploid cells. So a cell or organism consisting of one set of chromosomes, or just half, okay? The gamete sex cells are haploid. They only have 23 chromosomes. Which should make sense, right? If a person needs 46, you're going to get 23 from your mom and 23 from your dad. Exactly. So we symbolize this as N because you have just the number of chromosomes. They're not doubled. Okay? When you think haploid, think half. Okay? Cool with that? All right. Mitosis versus meiosis. So in mitosis, which we've already covered, an important step of cell division in which a new nucleus is created occurs right before cell division, right? So mitosis only takes place in somatic cells. We see here is the original DNA. It gets duplicated, and then half ends up in that baby cell, half ends up in this baby cell. We're good with that, right? In meiosis, it's a process that occurs in gametes, in which the number of chromosomes are cut in half. So this time, you have your original DNA. It becomes replicated, okay? But this time, it is going to be cut in half, and then in half again. That double division reduces the number of chromosomes, okay, in each thing. Otherwise, if you got 43 from your mom and 43 from your dad, then you would have, what, like 86? Thank you. And then the next generation, if they got 86 and 86 chromosomes, it would just be like our cells would be so full of chromosomes you couldn't survive. So remember, meiosis is double division, okay? So cell division of the gametes, it halves the number of chromosomes, it occurs in all sexually reproducing organisms, and there are two stages of meiosis, or there's two divisions, meiosis 1 and meiosis 2. Oh, sorry guys. So meiosis begins with a germ cell, which contains 46 chromosomes. Can you go back? I'll go back, sorry. So meiosis begins with a germ cell, which contains 46 chromosomes. Okay, so it starts with a normal diploid cell, but it ends with 4 gametes, which each contain 23 chromosomes. So parents can produce many types of offspring, right? Families generally have similar resemblances, but you don't look identical unless they're identical twin, right? So there's obviously some variation that's happening within your family, okay? So every cell has a nucleus, and every nucleus has chromosomes. And the number of chromosomes depends on the species. For example, humans have 46 chromosomes. Chickens have 78. Frogs have 26. Peas have 14. It's totally random, okay? So diploid versus haploid. So when the cells in your body have a complete set of pairs, it's called diploid. When they have gametes, which are eggs and sperm, only have half, it's 23, and we call it haploid. I know I'm kind of rehashing this information over and over, but I just want to make sure that you get a good handle on it, okay? So why bother with meiosis if it's all this double division, you have to go from diploid to haploid, what's the point? Okay? When the gametes combine, the zygote or fertilized egg gets half of its chromosomes from mom and half from dad. Okay? That's why you have to give only half to your offspring. So zygotes are diploid, which means the two halves come together to make a full, right? Half the chromosomes from the sperm meet half the chromosomes in the egg, and then you get a little baby, and it's so cute, and then it grows up, then it gets less cute for a while. You know how it goes, right? So the chromosome number of the egg and sperm is cut in half to ensure that the zygote will have the correct number of chromosomes. So meiosis, like we've said, is the process of creating a gamete or sex cell. It is similar to mitosis, but instead it will produce 4 non-identical daughter cells that are each haploid. Okay, so here we can see mitosis. If that was your skin cell, it would divide. You have 2 new skin cells. In meiosis, the diploid cell divides. It divides again, and it becomes 4 haploid cells. So remember we talked about genes are located on the chromosomes, and genes control an individual's traits such as blood type and eye color, and chromosomes come in matching sets called homologous chromosomes. So, for example, you have 23 pairs of chromosomes, right? You get chromosome number 1, you get a copy from mom, you get a copy from dad. That's why you see these paired up in color with each other. So the red ones are a set, the green ones are a set, the blue ones are a set. That's kind of what it's trying to show you. We call this a karyotype, and it's a picture of an individual's chromosomes. The color-coded pairs of chromosomes you see are called homologous pairs. Good. So homologous chromosomes, what do you need to know about these? You don't need to know all of these details, but I want to give you a little more information so the really important stuff sticks, okay? So chromosomes that are paired during meiosis, they pair at the synapse. They consist of 4 chromatids total. They only present during meiosis, and one pair is inherited from the father, the other pair from the mother. They're identical in shape, size, and gene location, and during meiosis, they swap genes in order to diversify gametes. So, when this process is happening, it's going to further mix up all the genetic variation, which is why your siblings look similar to you, but they're not identical to you, right? It's because of this crossing over. So there's moms, there's dads. Okay, here's what crossing over looks like. So a process in which homologous chromosomes exchange genes. The chromosomal points at which crossing over occurs is called a chiasma, which is a weird name, I know. Okay? It is important for genetic variation. If not for crossing over, gametes would be identical. So all of your siblings would be identical to you. It would just be like mini clones, like a little army. Okay? And this occurs during prophase 1. So you can see from this picture, right? Your brother or sister might have genes like this. You might look a little different than them because crossing over happened. So instead of just getting a red and a green, you get a green-red and a red-green. Okay? We'll get a little bit more in detail from that, but it's just supposed to show you that it increases genetic variation. So oogenesis. Oogenesis is the process of making eggs in females. Okay? During oogenesis, only one cell develops into an egg. So technically it should be making 4 different gametes, right? But in females, we make 1, and the other 3 we consider waste and call them polar bodies, and they die off. We spend our energy making 1 really good egg. Okay? In spermatogenesis, meiosis in males makes sperm. And so we call that spermatogenesis. Genesis meaning making in the beginning. Sperm. Very exciting. So we know meiosis is divided into 2 stages. So prophase 1 of meiosis, the homologous pairs form. The chromosomes trade genes if they need to, so crossing over can occur here, and just like we looked at in the last slide. Then it goes to metaphase 1, anaphase 1, telophase, and cytokinesis 1, and then it does it all over again. But the second time it divides, there's no replication of the DNA. So that's where you're seeing it cut in half in terms of the number, okay? We'll skip that. So we already talked about the steps of meiosis. This is clearly in your book for you too. just so you know. I'll play this for you in a minute. So let's look at this together real quickly. Meiosis involves how many divisions? You get 4 gametes, but how many times does it divide? 2. Very good. Okay? The divisions in which one diploid germ cell divides into how many haploid cells? 4. Very good. So meiosis is the cell division of gametes in which the number of chromosomes is halved. In mitosis, the cells are identical. In meiosis, are they identical? No. Why? There's 2 reasons why. Okay? The first one is it's only half of chromosomes, right? Is that the ugliest writing you've ever seen? I'm so sorry, guys. And the second reason is, what was that thing we learned about that increases genetic variety? Yes. You guys are smart. Crossing over. This type of question you will see on the test. Okay? Are we good? All right, so that's it for the notes.